The speaker for the third plenary is Professor Dan Kuritzkes. Professor Kuritzkes is the Harriet Ryan Albee Professor of Medicine at Harvard Medical School and Chief of the Division of Infectious Diseases at Brigham and Women's Hospital. He has published extensively on antiretroviral therapy and drug resistance in HIV infection. He previously chaired the AIDS Clinical Trials Group and served as a member of the NIH Office of AIDS Research Advisory Council and the U.S. Department of Health and Human Services panel on guidelines for antiretroviral therapy. He is going to give us an update on new antiretroviral therapies. Thank you, Professor Kuritzkes.
Thank you very much, Denise, and thank you, Professor Prapan and the committee for inviting me to speak at the 22nd meeting. It's really a great honor to be here. I'm going to give you an update on some of the new drugs that are being developed for antiretroviral therapy, so we'll be looking now into the future. Let me begin with my disclosures. I consult for several of the companies involved in the development of these drugs. Now, we know that there are more than 30 different entities that are currently approved by the U.S. Food and Drug Administration and by many other regulatory bodies around the world for the treatment of HIV infection, and these fall into various classes. The drugs listed in black are approved for initial therapy as well as subsequent lines of therapy, and the ones shown in the lighter color are approved only for the use either as switching therapy or in treatment-experienced patients. So one could well ask, given the wealth of drugs that we have and their extraordinary efficacy and generally good safety and tolerability, do we really need more drugs? The current regimens that we have are highly effective. They're generally safe and well tolerated. They're all or nearly all once daily, so highly convenient as single tablet regimens. And particularly with the advent of drugs like dolutegravir and bictegravir and with the boosted protease inhibitors, they have high barriers to resistance. But there are still concerns that persist, and these concerns regard the long-term and unanticipated toxicities. Who would have thought that integrase inhibitors might be associated with substantial weight gain, for example? There's the desire for even less than daily dosing in terms of frequency. And there are lingering concerns about the emergence of resistance. The world is about to embark on an enormous experiment of switching from efavirenz-based to dolutegravir-based therapy, as you just heard from the previous speaker, and what that will mean in terms of potential for emerging drug resistance, especially in lower income countries where switches may occur without appropriate virologic monitoring, is something that is of great concern to the WHO and other bodies. So there's always the need for new drugs, and in particular for the need for longer-acting antiretrovirals. These would allow for less frequent dosing that could improve adherence to these regimens, and it could improve the effectiveness of antiretroviral therapy. Because with less frequent dosing, the potential to provide drugs as directly observed therapy becomes much more of a reality than if you need daily dosing. And so there are several different approaches that are currently in development, and I'll focus my talk on these three drugs this morning. These include the use of injectable cabotegravir and long-acting rilpivirine, islatravir, which was previously known as MK-8591, which is a novel class of drug known as a nucleoside reverse transcriptase translocation inhibitor, and the novel class of capsid inhibitors, GS6207. I'll speak on Friday as part of my talk on managing drug resistance about some of the other newer drugs that have either been recently approved or are awaiting approval as viral entry inhibitors. So let's turn first to a discussion of long-acting cabotegravir plus rilpivirine. Both of these drugs have been formulated as nanoformulations that allow either subcutaneous or intramuscular administration, leading to very long half-lives of these drugs. And you can see here for cabotegravir, the scale here is in weeks. that in the open figures, the intramuscular injection, which is what's been moved forward into the phase 3 trials and is now pending FDA approval, that with injection you can have a half-life that lasts for 26 weeks. And so this is after a single injection. With rilpivirine, here you see with multiple injections, you can sustain trough concentrations that are substantially above the protein-adjusted EC90 at monthly intervals. And so based on these data and based on the LATTE series of studies, 2 large phase 3 studies were organized, and the results of those studies have been presented, and I'll summarize them for you here. The first of these was the ATLAS study, and this study looked at participants who were already on a suppressive antiretroviral regimen. That regimen could have comprised any of the major classes, protease inhibitors, non-nucleoside RT inhibitors, or integrase inhibitors, along with a 2-nucleoside backbone. And the candidates were randomized equally to either continue on their already prescribed therapy or to have a 4-week lead-in of orally dosed cabotegravir and rilpivirine in order to exclude the possibility of any hypersensitivity reactions, because once you inject the drug, it's very hard to remove it. And then if the drug was well tolerated, then after 4 weeks, they switched to monthly injections of cabotegravir and rilpivirine with a 48-week endpoint, after which in this extension phase, all the participants were offered the opportunity to continue with monthly dosing of cabotegravir and rilpivirine, or to receive the drug every other month, because additional data had suggested that once every other month dosing might be just as good as every month dosing. This slide shows the primary and secondary endpoints of the study. As a switch study, the primary endpoint was really the number of failures that occurred, and then the secondary endpoint was also the overall success of maintaining virologic suppression. And so here you can see that in the primary endpoint, there was essentially no difference at all between, in the failure rate between those who remained on their previously prescribed combination antiretroviral regimen and those who switched to the injectable cabotegravir and rilpivirine, meeting the non-inferiority margin. And in the secondary analysis, looking at the success rate, you can see that between 93 and 96% of participants maintained virologic suppression. This was likewise met the non-inferiority margin, and so this study was a substantial success. If we look at adverse events, the regimens were well tolerated. There was, in terms of any adverse events that occurred in at least 10% of participants, there was some upper respiratory infections and nasopharyngitis, some headache, and as far as drug-related adverse events were concerned, there was a little bit of fatigue, pyrexia, headache, and nausea in the injectable group compared to none in the continued group. These were people who had been tolerating their regimen for many months already, and so it's not surprising that there were no new adverse events. Very few participants had adverse events that required withdrawal. Now, the big question with this form of therapy, of course, is how well tolerated were the injections? And this slide summarizes the injection-related adverse events. You can see that there were nearly 7,000 injections given during the study up to the primary endpoint, and that approximately 20% of these injections were associated with injection site reactions, the main adverse event being that of pain. So about 83% of the adverse events associated with injection were local pain at the injection site. It was much less common for there to be nodules or swelling, sort of a true enfuvertide-type injection site reactions. And notably, the duration of these reactions was only 3 days as a median, and only 4 participants withdrew from the trial as a result of injection site reactions. The partner study was FLAIR. This study had a slightly different design. Here, treatment-naive participants were recruited into the trial, and they were initially placed on a regimen of dolutegravir, abacavir, 3-TC as a fixed-dose single-tablet regimen, and those who achieved full virologic suppression could then be randomized to either continue on that single-tablet regimen or switch to oral cabotegravir and rilpivirine for a lead-in period, after which they then received the injectable, and again with the primary endpoint at 48 weeks and follow-up to 96 weeks. So here there's a direct comparison between injectable cabotegravir and orally dosed dolutegravir. Like the ATLAS study, this study met its primary endpoint of non-inferiority, showing that there was, again, virtually no difference between the arms with respect to the rate of virologic failure and nearly identical rates of maintained virologic suppression at 94% and 93% for the orally dosed and injectable arms, meeting the non-inferiority endpoint. I won't summarize the safety data from FLAIR, which looked almost exactly like the ATLAS data for the sake of time, but want to highlight some rather curious findings with respect to virologic failure and resistance. There were 6 participants in this between ATLAS and FLAIR who had virologic failure and developed resistance to cabotegravir. And curiously, all 6 of these individuals were people who have a subtype A virus. But it's not just any subtype A, it's actually an A6 isolate. These slides show this as being an A1 isolate, but that's actually incorrect. More careful sequence analysis confirms that these are actually A6 viruses, the viruses that are, or the subtype that is predominant in Russia. And you'll notice that 5 of the 6 individuals here are of Russian nationality. There's 1 person in France who had an AG recombinant virus. And what is particularly striking is that the A6 subtype has as a polymorphism at baseline a substitution at position 74 in the integrase gene, and all of the viruses in which resistance occurred carried this 74 substitution. You can see there's a variety of resistance mutations in integrase that emerge at the time of virologic failure, including the 155 mutation here, and then here a 148, a 140, and another 148. It's not yet understood what the role of the 74 polymorphism is, if it has any role at all in potentially enhancing the emergence of resistance. The 74 mutation by itself has no effect on susceptibility to cabotegravir or other integrase inhibitors. And in collaboration with ViiV, our laboratory is now attempting to analyze whether there is a fitness advantage of viruses that carry the 74 mutation that promote the emergence of other resistance mutations. There may be other factors involved in the failure, including body mass index. Several of the participants had rather large body mass indices, and whether drug was appropriately delivered intramuscularly as opposed to subcutaneously. All of the individuals who had virologic failure had drug levels that were in the lowest quartile of drug at the trough, but there were many other people who had similar drug levels who nevertheless remained suppressed. So there's a lot to still understand about these virologic failures. This drug combination is pending FDA approval. It was expected to be approved by the end of December, but approval was upheld because of some regulatory concerns around manufacturing issues that need to be resolved, and so hopefully that will happen in the current first quarter of this year, and we'll see this agent approved relatively soon. Another long-acting drug that is further behind in development, but is now has just completed phase 2 and is moving into larger expanded trials, is the NRTTI islatravir that has previously been known as MK8591 and EFDA, or ethinyl fluoroanyl deoxyadenosine. This drug has actually been around for a very long time. It's been studied by Mitch Mitsuya and Peter Sarafianos and Mike Parniak for nearly 20 years, actually, but had been put aside when there was a lot of concern around toxicities of the nucleoside reverse transcriptase inhibitors, particularly because this was a fluorinated version of these drugs. Islatravir has two unique properties. One is the presence of this ethynyl group, and the other is the fact that it actually has a 3' hydroxyl. So this drug is not a chain terminator in the classic sense. It can accept another nucleoside, but it prevents the reverse transcriptase from moving along the template and therefore is called a translocation inhibitor. It is a highly potent agent with IC50s in peripheral blood mononuclear cell culture of less than 1 nanomolar. Whoops. I mentioned doravirine in passing here. This is already an approved drug, and I bring it up only because the phase 2 study of islatravir is done in combination with doravirine, a next-generation NNRTI that has a unique resistance profile, is not an inducer or inhibitor of the cytochrome P450 system, and can be dosed once daily without regard to food. This slide shows the design of the phase 2 dose ranging study that combined islatravir with doravirine. Ideally, this would have started as a 2-drug study, but the FDA insisted that the study begin as a 3-drug study and then allowed a switch to 2 drugs once participants were virologically suppressed. So in this trial, the comparator arm is a fixed-dose combination of doravirine 3TC and the TDF formulation of tenofovir, and then the experimental arms looked at 3 different doses of islatravir, initially in combination with doravirine and 3TC, and then after 24 weeks dropped the lamivudine and continued for the next 24 weeks with islatravir and doravirine alone, again compared to the 3-drug regimen, and the plan being after week 48 to move forward with a selected dose for an extension phase during maintenance. This slide shows the virologic outcomes at week 48 by FDA snapshot analysis, and you can see that between 77 and 90% of the participants achieved and maintained virologic suppression. There were slightly more failures at the highest dose of islatravir, but these numbers, given the relatively small sample size, are not statistically significant. And here you see the log drop during the initial 24 weeks and then the sustained virologic suppression out to week 48. In terms of adverse events, there were relatively few. There were no deaths. There was a single serious drug-related adverse event which actually occurred, that resulted in discontinuation, that actually occurred in the doravirine 3TC tenofovir arm, not in the islatravir arms. 2 participants in the highest dose group for islatravir discontinued due to AEs, one for GI side effects and another who had reactivation of hepatitis B virus infection. And there were somewhat more drug-related AEs in those who were receiving doravirine compared to those at any of the doses of the investigational drug in this trial. This is a duplicate, I apologize. So how might this drug be administered? Well, islatravir in these studies was given orally, and it is being investigated for potential weekly oral dosing, but a much more exciting approach is to provide it as an implant. Here you see an example of implants as they're currently used for long-acting contraception with implantable etonogestrel. And this is the size of the implant, and the implant is an automated device that can be done by allied healthcare professionals, doesn't need a surgical implantation by a physician. And we heard at the Mexico City meeting last summer preliminary data looking at the pharmacokinetics of islatravir in seronegative study participants who received one of two doses, either 54 or 62 milligrams of islatravir as a single agent. And here the orange lines, the heavy line shows you the average intracellular islatravir triphosphate concentration. So this is the concentration of active drug during the 12 weeks that these implants remained in place. And the dotted lines show the minimum and maximum of the range. And then you can see at the higher dose, everybody, even at the minimum, was well above the threshold of 0.05 picomoles per million cells, which is the amount needed to assure virologic suppression. And in modeling studies based on these 12-week data, it's projected that at week 52, 1 year of having the implant in place, you would still have concentrations of drug at about 0.076 picomoles per million cells, which is sufficient drug to inhibit HIV replication. This by itself would be an astonishing advance for prevention. You would need a partner drug in order to create a regimen in order to be able to have an implantable antiretroviral regimen, but the possibility that we could be giving implants once or twice a year to sustain virologic suppression is really now only a couple of years off. The last drug I'll speak about, which is yet again a little bit earlier in clinical development but moving rapidly forward, is the capsid inhibitor GS6207, which thus far does not have an official chemical name. And just to remind you about the function of HIV capsid, capsid is what we also know as P24, the major protein of the virus particle itself. The capsid self-assembles during the process of virus production and then has to disassemble upon virus entry. And the capsid inhibitor binds to the capsid monomer in a way that both prevents the assembly of the capsid and prevents its disassembly. And that's important because it means that the drug acts at several different stages in the virus life cycle. It interferes with capsid disassembly and therefore interferes with nuclear transport of the capsid reverse transcriptase and integrase complex following reverse transcription of the double-stranded DNA into the nucleus so that integration cannot happen. Therefore, capsid acts on early stages of the virus life cycle, but it also prevents virus production by inhibiting the appropriate formation of the capsid. And so it has the capacity to prevent production of virus from cells that are already infected and therefore is unique in this dual function in the virus life cycle. A phase 1b study was recently reported at the Mexico City meeting and updates presented at the EACTS meeting in Basel a couple of months ago. This study examined the activity of 3 different doses of GS6207 given as a single subcutaneous dose. After patients were followed, or participants were followed for 10 days, after which they all switched to a fully suppressive regimen in order to sustain the virologic suppression without risk for emerging drug resistance. And you see here in this slide the slope of the viral decay, which showed a very nice dose-proportionate response with about a 2-log decrease being achieved at day 10 in the highest dose. This is a really very potent agent if you look at the amount of drug that's being given here. The safety data are still blinded, but looking at the blinded data, there were some injection site reactions, not very surprisingly, and some upper respiratory infections, a little bit of nasopharyngitis, but all of these adverse events were grade 1 and 2 in severity, so at least in this small preliminary study, there were no serious adverse events and no discontinuations due to adverse events. So I mentioned that there are other drugs in development. Actually, ibilizumab, which is a novel drug, is approved, and I'll say a bit more about that on Friday as part of the management of patients with extensive drug resistance. Fostemsavir is another entry inhibitor that is pending approval. Leronlimab, which has been around for many, many years as Pro-140, is likewise pending approval, and in preclinical development is a maturation inhibitor that is yet to enter into clinical trials. So we really have remarkable promise for new agents that have the potential for revolutionizing our approach to antiretroviral therapy. And I think that as successful and as incredible the advances have been with once-daily single-tablet regimens, being able to look forward to the advent of injectable or implantable therapy offers enormous opportunities for both prevention and treatment going forward, and I expect that these agents will be approved within the next 3 to 5 years, and for cabotegravir, rilpivirine in the next 3 to 5 months. So let me end there, and thank you very much for your attention, and I'll give the floor back over to Denise. Thank you.